Hello everybody, and welcome to today's webinar entitled Take Control of Your Flow Cytometry, a comprehensive guide to the essential controls in flow cytometry. Your host will be Mike Blundell, one of our product managers here at Bio-Rad. This is the second in our series of flow cytometry webinars, with more to follow. Mike graduated from the University of Edinburgh with a BSc in immunology. He then moved to the University College London, where he joined the group of Professor Adrian Thrasher and obtained his PhD. His thesis research was focused on the primary immunodeficiency Wiskott-Aldrich syndrome. He investigated novel activating mutations and developed lentiviral vectors for use in gene therapy treatments, some of which have now been used in clinical trials. Mike contributed to 50 peer-reviewed papers and has now left academia to become a product manager for flow cytometry. As we are trying to make this webinar as interactive as possible, please submit questions or comments via the Q&A box located at the bottom of the screen. We will try to answer as many questions as possible in the session following the main presentation. Should any questions remain unanswered, we will reply by email. The webinar will be recorded and an on-demand version will be available shortly. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide, which describes solutions for the most common technical problems. In addition to webinars, we regularly post blog articles, not just on flow cytometry, but other applications and research areas. We also have blog posts on current scientific discussions and the latest biomedical discoveries. Please feel free to check out the blog and comment on one of the many articles. In addition, you can follow us on social media to stay up to date on antibody resources, news and products. Our social media channels can be accessed via the icons at the bottom of the screen. During the webinar, you can interact with us on Twitter by submitting questions or webinar feedback using the hashtag Bio-Rad-Webinars. I hope you will enjoy the webinar, and now over to you, Mike.
Hello everyone, and welcome to this flow cytometry webinar on the subject of controls. Controls are not necessarily the first thing that comes to mind with flow cytometry. The urge to see the dots appear on the screen, get the data, and the speed at which results can be obtained can sometimes overtake some of the careful planning that needs to be done. With this in mind, this webinar aims to take you through some of the essential controls that should be part of any flow cytometry experiment and that will help you get the most from your data, whether this is one colour, 7 colours as shown on this slide, where I have stained human blood for CD3, 4 and 8 to look at T cells, CD19 and 20 for B cells, and CD14 and CD66B for myeloid cells, or even for those very experienced users who are doing 17 colours. So the first control I want to mention is the simplest, the unstained control. You should have an unstained control for each different cell type or experimental model in your experiment. Once you have identified your cells by their forward and side scatter characteristics, for antibody-labeled cells, you'll need to check the fluorescence levels. An unstained control will allow you to see your negative population and show how much autofluorescence you have. You can then set your voltages and detectors to move the cell population towards the y-axis to allow a larger dynamic range of fluorescence to be accurately measured. This will also give you an idea of where to set your negative gates, although this may change depending on your experiment, which we will discuss later. The next control to talk about is the FC block. Some cells, such as NK cells, B cells, dendritic cells, and macrophages, have FC receptors on their cell surface. FC receptors allow antibodies to bind via the constant region of the antibody, leading to increased background staining. Pre-incubation with an FC blocking antibody for 10 to 15 minutes will prevent binding of other antibodies via the FC portion. You can then continue with your antibody, making sure you do not wash off the FC block, thus ensuring you get specific antigen staining. As can be seen in the overlaid histogram, THP1 cells, a human monocytic cell line, which has FC receptors, were stained with CD11A FITC. Without an FC block, there is no apparent difference between the isotope control in red and the CD11A antibody in blue. However, when an Fc block is added, there is a clear difference between the isotype control and the CD11A antibody. The third control to discuss is the viability dye. Dead cells have increased autofluorescence and increased binding of antibodies through non-specific interactions. The result of this is that the negative population may often be more positive, so weakly positive cells and rare cells may be masked by the negative population. In addition, cells may appear falsely positive for a marker. Often, forward and side scatter gating is used to exclude dead cells, but not all will be excluded. Therefore, inclusion of a viability dye is important to exclude these dead cells. There are two main groups of viability dyes. The first we're going to discuss are the nucleic acid binding dyes, such as propydium iodide and 7-AAD, which are excitable by the 488 and 561 lasers, and DAPI excited by the 355 laser. When these dyes bind to nucleic acid, their fluorescence characteristics change. They are excluded by live cells as they are not membrane permeable, but dead cells, which have a compromised membrane, will be stained. Bio-Rad's Ready Drop formulation is available as a convenient, ready-to-use solution, which is stable at room temperature. Simply add 1 to 2 drops directly to samples after being stained with antibodies, then acquire as normal. The dead cells can then be identified and removed from the final analysis. As these dyes rely on membrane integrity, it is not possible to fix the samples. The second group of dyes available to enable you to discriminate dead cells from your live cells is the protein binding dyes. As can be seen in the cartoon, these dyes will bind to both live and dead cells. However, when a cell has a compromised membrane, as seen in dead and dying cells, there is access to a greater amount of protein. Therefore, they will have higher fluorescence. An example of this can be seen in the FACS plot, where Jurkat cells have been stained with one of our VivaFix dyes that is excited by the blue laser and emits at 521 nanometers. The benefit of these dyes over the DNA binding dyes is that once stained, the cells can be fixed without any reduction in the resolution between live and dead cells. They are also available in a range of excitation and emission spectra from UV to far red, so you have a convenient addition to multicolor flow cytometry panels. An example of how important viability dyes can be is shown on this slide. In the top panel of human peripheral blood, a gate on the forward and side scatter has been used to exclude debris and dead cells. However, there appears to be a population of CD14 positive cells, some of which are CD3 positive, shown in the red circle. When propydium iodide is used to exclude the dead cells, this population disappears. So the next topic I want to discuss is one that can divide researchers, the isotype control. Isotype controls were developed to control for unwanted background staining due to antibodies binding to targets other than those intended when performing surface staining. The antibodies are raised against irrelevant antigens not found on your cells of interest. Often this includes targets such as KLH or DNP, not found on mammalian cells. They therefore show the amount of binding that is due to low affinity interactions, Fc binding, or binding due to the fluorophore. The most appropriate isotype control to use will match the specific antibodies used in your experiment as closely as possible. This means the host species, the immunoglobulin subclass, the fluorophore, and the amount of fluorophore conjugated to the isotype antibody should all match your primary antibody. An example is shown below, where J774 macrophages were stained with CD11B and F480, with the appropriate isotype controls included to determine specific binding. As can be seen in the plots, on the left-hand side, without an Fc block, there is isotype binding shown in the red circle. In the middle plot, when an Fc block is used, this disappears and allows you to have confidence in the staining provided by the primary antibodies as seen in the right-hand side. As the fluorophore conjugation to the antibody can vary between suppliers, it is best to purchase the isotype control from the same supplier as the primary. An isotype control with less fluorophore conjugated may not show all of the background staining. They should also be used at the same concentration as your primary antibody, so the level of non-specific binding should be the same. If you are unsure of the level of fluorophore binding, the fluorophore protein or FP ratio can easily be calculated in 4 easy steps. Firstly, ensure all excess fluorophore is removed. Then determine the concentration of protein by measuring the absorbance at 280 nanometers and divide this value by the extinction coefficient of the protein. Then determine the fluorophore concentration by measuring the absorbance at maximal excitation. For example, for FTSE, use 495 nanometers. And then divide this value by the extinction coefficient of the fluorophore. Finally, divide the two values to get the FP ratio. For more information, go to www.Bio-Rad-antibodies/FP ratio. We also have an Isotope Controls webpage where you can find information and search for the right isotype to match your primary antibody. As I mentioned earlier, isotype controls can be controversial, and their use often divides researchers. If you want to use isotype controls, and many journals will require them before publication, here are some tips for using them. Remember, they've been optimized for surface staining, and they may not be the best control for intracellular staining. They should not be used to determine compensation levels or the negative population. They are there to determine the background staining. Finally, they should always be used in conjunction with other controls, which we'll discuss later. There is another control which can help determine specific binding, and that is the isoclinic control. The isoclinic control is where cells are stained in the presence of an excess of an identical unlabeled antibody. As we can see here in the figure, regular staining is shown on the left-hand side. On the right-hand side is the inclusion of the isotonic control. As the unlabeled antibody competes with the labeled antibody for specific binding, the only binding will be seen via the fluorophore. If there is no fluorescence, then the fluorophore does not bind. Finally, if you want to read more on isotype controls, I have selected a few publications here which will help you. Intracellular staining can be tricky, with high levels of background often seen. Isotype controls, as I mentioned, are not always suitable, and if a secondary is used, using the secondary antibody alone can often reveal some of the non-specific binding. In addition to an unstained sample, appropriate controls for intracellular staining can include antibodies known not to bind to your cells, or staining cells you know lack the antigen to reveal the background staining. Ideally, this should be done with the same type of cell. If you are doing surface staining in addition to intracellular staining, you can perform this without the intracellular staining, and staining for the intracellular antigen without permeabilization will also give you some information on the background staining. In addition, for controlling for the antibodies, appropriate controls for the experiment must also be included. The fixative and permeabilization buffers will affect your experiment, so may need optimizing, as will the choice of fluorophore. For example, large fluorophores, such as PE and APC, may not be appropriate for nuclear staining and can be affected by alcohol fixation. In addition to controls for the antibody binding, there are also important biological controls you'll have to think about for your experiment. These controls are often used in intracellular staining, but are also applicable for rare or novel targets, where you may be unsure of your expected result. Firstly, it can be really useful to include a known negative or a known positive in your experiment. This will allow you to gain some insight into the staining patterns and the level of background you might expect. This can be done simply using cell lines or primary cells with known expression patterns taken from the literature, or by specifically knocking down the protein in the correct cells using siRNA or CRISPR technology. When looking at markers or cytokines which are increased or decreased in response to a stimulus, you should always include your negative and positive controls. Similarly, if possible, a fully stimulated control should be included to show the dynamic range. For example, if you are looking at cytokine release in T cells, often PMA plus ionomycin is an example of a fully stimulated control. but this will be different depending on the cells in each experiment. When performing multicolor flow cytometry, single stain samples are essential to determine the levels of compensation that may be required. Although a fluorophore, such as FITC, has a maximal excitation at 525 nanometers, it also emits fluorescence at higher wavelengths, so there will be some photons with higher wavelengths that will potentially pass through other filters and will be detected in other photomultiplier tubes, or PMTs. An example is shown here, where FITC and PE are combined. The FITC emission is shown in green, and the PE emission is shown in yellow. The filters used in the cytometer are shown in grey. In red is the percentage of FITC that spills over into the PE detector. So how do you determine the amount of spillover? Well, this is easily done with single staining for each of your fluorophores in your experiment. Single staining will reveal the level of spectral overlap between different fluorophores and allow you to remove or compensate for this overlap. An example can be seen here in this, where single staining has been performed for CD45RA FITC on human peripheral blood T cells. There is a tail of staining that appears to also be positive for PE. When compensation, in this case removing FITC from PE, is applied, the true staining is shown. Important things to consider when using single stain samples for compensation are that the fluorophore needs to be the same and its brightness similar to the one used in your experiment. You also need to collect enough events to accurately apply the compensation and be statistically significant. Finally, as mentioned earlier, each fluorophore should be compensated against every other fluorophore used, and single stains will be required for every fluorophore used. So the final control I will discuss today is the fluorescence -1, or FMO control. Like single stain controls, FMO controls are also important when building multicolor flow panels, as they will help you determine where to set your gates. This is important to be able to determine the positive from the negative, and particularly important for low expressing antigens. When acquiring data, there is fluorescence spread, especially from brighter fluorophores. The spread is derived from measurement errors and is seen more in longer wavelength light. This can be more noticeable after compensation, as compensation amplifies errors in the data. The fluorescence -1 control, as the name suggests, is the inclusion of all the stains in your panel with the exclusion of one of them. This then allows you to accurately assess the effect of all the other fluorophores on the left out parameter, particularly the spread. An example is shown below in a 4-color panel, where the spread into the PE Cy5 channel can be seen. An unstained control is not sufficient alone to set the gate boundary, shown by the red line. As can be seen in the middle panel, the FMO shows there is a fluorescent spread into the PE Cy5 channel. This allows a more accurate gate to be set, and the true positives can be seen in the final panel. A dim expressing population could be missed without the FMO control. It is important to perform an FMO control for all of the fluorophores in your panel when starting a new experiment, as it will allow accurate setting of gates. However, it is best to avoid spread as much as possible in your experimental setup by avoiding spillover and therefore compensation and data spread into channels requiring high sensitivity. So in summary, today I have shown you the various controls that are important in flow cytometry experiments. These include unstained samples, inclusion of FC blocking, and viability stains in your antibody staining protocols. I have discussed isotype controls and what they should be used for. I then discussed the various intracellular controls and biological controls that should be considered. And finally, I went through the controls that could be used when building multicolor panels, specifically single stain compensation controls and FMO controls. Remember, controls are important in all experiments, and using these controls properly should help you make the most of your flow cytometry data. So as we're reaching the end of this controls webinar, I'd just like to bring to your attention the various flow cytometry resources available on bioradantibodies.com. As well as information on controls discussed today, we have tips and tricks to build multicolour panels, gating strategies, fluorescence charts, and resources on immune cell marker selection to help pick the right antibodies. We also have guides and resources for other antibody-based applications, such as Western blot and microscopy. In addition to this flow cytometry webinar on controls, if you go to Bio-Rad Antibodies/antibody videos webinars, you'll be able to view on demand additional application-based webinars and research area webinars. And so the last slide today is to notify you of the new ZE5 cell analyzer. This innovative new cytometer has the flexibility to meet a broad range of needs, whether it is 2 parameter or 28 parameters, from single tubes to 384 well plates. The ZE5 is accessible for novice flow cytometry users with a fluorophore selector, spectra viewer, and quick action buttons to select the next setup step, and yet is flexible enough for the most experienced users, with extra forward scatter detector for small particles, absolute counting without beads, and the ability to add reagents to your samples in real time for kinetic experiments. To find out more, go to bioradantibodies.com/ZE5.
Thank you, Mike. And now I would like to open the Q&A session. I am aware that some of you might have to leave us early. If you are unable to stay for the duration of the Q&A session or missed your chance to submit a question, you can contact us or our technical support team by email. We offer technical support in English, French and German, and you can simply call us or send us an email. For more information about how to get in touch with our technical support team, please visit our website, bioradantibodies.com/technical. For the Q&A session, we are joined by another member of our team, Dr. Steve Clasper. Steve holds a PhD degree in biochemistry from Liverpool University and has a wide-ranging experience in antibody applications that he gained in both academia and industry. And now, over to you, Steve.
Thanks, Kate. Thanks, Mike. Hi, I'm Steve, and I've been looking over the questions you've been sending in whilst Mike was presenting. I've selected a few that I think might benefit the wider audience, so let's get started. The first question is, how do I control for autofluorescent cells? The first step we suggest for autofluorescence is to try and use fluorophores at longer wavelengths. Autofluorescence is generally worse at the blue and the yellow end of the spectrum, so if you use fluorophores with a longer excitation and emission wavelength, you can reduce the effects. So APC, Alexa Fluor 647 or Alexa Fluor 700 would be a good starting point. If you do need to use fluorophores at shorter wavelengths, such as FITSI, you can distinguish true signal from autofluorescence by plotting the data against another compensated shorter wavelength fluorophore, such as PE. Genuine signal will only appear on the FITSI axis, whilst autofluorescence will be visible on both fluorophores. And the next question we have is, which viability dye is the best? Well, this really depends on what you're doing. If you don't need to fix your cells, then using a DNA stain such as PI, 7AAD, or DAPI is probably the easiest. However, if you do need to fix and or permeabilize your cells, then a protein stain such as VivaFix is what you need. If you're doing a multicolor panel, you may also find that the greater range of fluorophores in the VivaFix range are an advantage. Okay, there's a question here which is, can I include isotopes in my FMOs? And there's another related question, why can't I set my gates with isotope controls? I've put these together because they're quite interestingly related. Firstly, yes, you can actually include isotope controls as a negative channel in your FMOs. However, you should only use these to gauge the level of non-specific binding. Don't use them to set the gates. Which brings us to the question, why not? Well, it's because the binding of isotope controls is generally at a low level, whereas FMO controls for the spread of data seen at higher fluorescence levels. Setting gates with the isotope controls can therefore lead to false positives. There's another couple of related questions here. One is, how many cells should I collect? And another is, how many events do I need for compensation? Well, this depends on your cell type and your cell populations, but generally to analyze a population or compensate, we recommend between 1,000 and 5,000 events. Next question is, if there isn't an FC blocking reagent available for the species of cells I'm testing, what should I do? FC blocking reagents are usually antibody reagents that bind to the FC receptors such as CD16 and CD32, and they prevent them from binding other antibodies non-specifically. So for this reason, they're usually unique to one species or a small number of closely related species. If you're working with cells from a species that's not human or a lab animal such as mice or rabbits, it can be tricky to find a reagent. The usual recommendation is to block the FC receptors on your cells using a pre-incubation step with serum from the same species as your test antibodies, such as mouse serum for mouse antibodies. However, it can be more effective to use serum from the same species as your cells if you can get it. This is because antibodies from the same species as your cell will bind to the FC receptors with the highest affinity. An additional benefit over using a serum from the same species is that your antibody, it allows you to use a secondary detection antibody if required. A question here on titration. Despite titrating my antibodies and carefully setting up the compensation controls for them, I still sometimes see cells that appear to be double positive for unexpected markers. Are these real? Well, there's two steps to eliminating false double positives that you need to check before you investigate this any further. The first thing is to ensure that you have adequate FC receptor blocking in your assay, because the non-specific binding of antibodies can lead to false positives. So I suggest you try using an isotype matched non-specific control antibody to check this. And the other thing is to make sure you've gated out any cell doublets from your analysis, as you might find that your double positive cells may in fact be two single positive cells stuck together. You can visit our website for tips on doublet gating. Finally, can I avoid compensation? Well, for dual colour staining and small panels, it's possible to avoid compensation providing your cytometer has enough lasers and filter sets. We recommend using fluorophores that are excited by different lasers as a starting point, such as Pacific Blue, Bitsy, and Alexafluor 647. You can add a fourth colour by putting in PE Alexafluor 750 Tandem. Now have a look at our website, www.bioradantibodies.com/no-comp panels, for a range of panels for human blood phenotyping that use this strategy. Okay, I think we need to stop there. I'll just reiterate that if your question hasn't been answered here, we will get in touch by email to give you some suggestions. Please let us know if our answers have helped, or of course if they haven't, and we'll do our best to get to the bottom of your problem. I hope that Mike's talk has given you the information to incorporate the correct controls into your flow experiments, and it gives you the ability to have confidence in your flow data, analysis, and results. So it just remains to say thanks very much for listening, and goodbye.